Hi viewers, this is Dr. G. Manigandan working as Assistant Professor and Head PG and Research Department of Biotechnology, JJ College of Arts and Science, Autonomous, Pudukkottai. Now I am going to discuss about lymphoid organs in detail. This slide shows you what do you mean by organism. As we all know about the atom, so that is the very smallest unit of matter that have the properties of an individual element. These atoms combines to form a molecule, and molecules combines to form a macromolecule, and macromolecules combines to form an organelle, and this organelle produces cells, and these cells will form a tissue which in turn form organ, and then by organ system, say for example digestive system, all the organ systems combines to form an organism. Example, the human being. And this is the immune system. First of all, with the function. The function of the immune system is to protect the body from various diseases. It recognizes, attacks and destroy various kinds of pathogen. And about the components of immune system, it is cells, organs and tissues. And this is a classification of the lymphoid organs. We are classifying the organs into two types, primary and secondary. What do you mean by primary? It is otherwise called as central lymphoid organ. Why it is central lymphoid organ? In that organs only lymphocytes will be generated. Examples under the category are bone marrow and thymus. And next one is the secondary lymphoid organ. It is otherwise called as periparal lymphoid organs. Examples under the category are spleen, Peyer's patches, lymph nodes, etc. And what the functions of the secondary lymphoid organ? So in that organs only, the lymphocytes will be interacting with the antigens, so that antibody will be producing. So in the secondary lymphoid organs. Coming with the primary lymphoid organ, the best example under the category is thymus. This is a very large organ in infants, approximately 70 gram in size, but it atrophies with the age, and in adult it is 3 grams. It is bilobed organ and it is located nearing the heart and the lungs. Each lobe has two compartments, namely cortex and medulla. The outer layer, which is nothing but the cortex, which consisting of lymphocytes and epithelial cells, and the medulla, that is the inner portion. The lymphocytes, the epithelial cells, the macrophages, the same cells, what the cells present in cortex, the same can be present in the medulla, and it possesses an important specialized cells called Hazel's corpuscles. It's nothing but the clusters of dying cells, the function unknown. Some of the scientists they have find out that these Hazel corpuscles, so that is the specialized cells for the production of cytokines. And these are the functions of thymus, can play a vital role in early immune system development, and it produces a hormone called thymosin. It develops T cells and it promotes the growth of lymphocytes. It shrinks after puberty. This slide shows you the thymus and disease. So when you remove the thymus from the any organism, what do you call it is thymectomy, the surgical removal of thymus. So the when you remove thymus from the mice, and it lack the production of T cells, as well as it lack the cell mediated immunity. And another property of thymectomy, some of the human population, they may be affected with a Dye-George syndrome, that is because of the deficiency of T cells. It's nothing but the primary immune deficiency disorder which is caused by the deficiency of T cells. And then the next with bone marrow. So this is the primary lymphoid organ present in mammals, and the primary function of bone marrow is to produce B cells. And now coming with the functions of bone marrow. It will produce all the types of blood cells and destruction of old RBCs also, and it stores Fe, that is ion, from the breakdown of hemoglobin. So these are the functions of bone marrow, and this is the bone marrow in different ages. So look at the first one, the infant stage. It is totally red in color, so we are telling it is red marrow. And you look at the second stage, the child stage, and here yellow marrow it begins, and when the child becomes adolescent, and yellow marrow begins and it grows well. So 75% of the bone marrow will be replaced by the yellow marrow, and when the adolescent becomes adult, you see the difference, and the total bone marrow, it becomes yellow marrow. And now coming with the next lymphoid organ is bursa of Fabricius. In mammals, B cells will be produced only in the bone marrow, but in birds, bone marrow is absent. So instead of bone marrow, B cells will be produced only in the bursa of Fabricius. And this is the characteristic feature of the organ, bursa of Fabricius. So why it is called as bursa of Fabricius? Fabricius, the name of a scientist who first discovered the organ in birds. So it is called as bursa of Fabricius. A dorsal outpocketing of cloaca, and it is a primary lymphoid organ present only in the mammals where B cells undergo maturation. It increases in size up to 3 weeks of age and then undergoes regression. When you remove the bursa from the birds, it is called as bursectomy. And when the bursa is removed, it affects the HMI, that is humoral immune response. And the B cells derive their name because they mature only in the bursa. Primary lymphoid organs over, and now we are moving to the secondary lymphoid organ. So it is otherwise called as periparal lymphoid organ. It consists of two categories based on the capsule: encapsulated organ and non-encapsulated organ. In the encapsulated organ, best example is lymph node and spleen, and non-encapsulated, it is MALT, that is mucosa associated lymphoid tissue. So these organs serves as the site for the interaction of mature lymphocytes with the antigens. Spleen, this is the one of the example under the category of secondary lymphoid organ. This is the largest organ, and it is located in the upper left abdomen, and it has sinuses filled with blood, and it consists of two tissue types, which is white pulp and red pulp. What is the difference between the white pulp and red pulp? White pulp, a material which is part of the immune system, mainly made up of WBCs, and red pulp, it is made up of blood filled cavities and splenic cards. About the functions of spleen, first function is immune function, specific, that is specificity, the production of specific immunoglobulin, that is IgM, and non-specific, in that sense the production of any antibodies rather than IgM alone. So that if it is non-specific, it can able to react with all the type of microorganisms. And the second function is filter function. It captures foreign particles by macrophages. The first function is culling and pitting, and both the functions can remove the platelets, red cells, and any particles. And the next function is pooling, and it is up to 30 to 40 percentage of blood platelets. They are sequestered within the spleen. And the reservoir function in animals, especially the dog, up to 1/3 of the blood volume may be sequestered in the spleen during sleeping and return to the circulation on waking, and this does not occur in the human beings. And the last one is the hematopoiesis, the formation of the erythrocytes. Coming with the next secondary lymphoid organ, lymph nodes. Coming with the structure, it is an elongated and bean shaped structure. It is 3 centimeter long, and it is enclosed in a fibrous capsule with the extensions. So that is called as trabecula, that incompletely divide the interior into the compartments. And about the functions of lymph node, can play a vital role in the defense mechanism of the body, and they can filter out microorganisms. And the major functions are lymphopoiesis, the filtration of lymph, the processing of antigens, the multiplication of the B cells and the T cells, and the antibodies produced or carried out to circulation, that is indirectly helping to mount an immune response. And the next secondary lymphoid organ is malt. The expansion for the malt is mucosa associated lymphatic tissue. Pear patches, tonsils, and other accumulations of lymphoid tissues are under the category of malt, and it acts as a guard to protect the respiratory and the digestive tracts. First of all, with the tonsils, and these tonsils are found at the opening of the throat. About the function, it protects the openings of the oral cavity and throat from harmful materials entering. There are 3 types of tonsils: palatine, pharyngeal, and lingual. Palatine, located at the sides of the throat, and pharyngeal, located nearing the internal nasal openings. And when it becomes enlarged, it is called as adenoid. Lingual, on posterior surface of the tongue. Tonsillitis. It is an inflammation of the tonsils, mostly commonly caused by the viral or the bacterial infections, affecting only in the children's. The symptoms may include sore throat and fever. The next organ is Pear patches. Why it is called as Pear patches? A patches found in the intestine, and that was discovered by the scientist Pear. So it is called as Pear patches, and it is compared with the tonsils of intestine found in the wall of small intestine. About the function of the Pear patches, it capture and destroy the bacteria in the intestine. Appendix. A function not fully known, but thought to provide a safe place for the bacteria, good bacteria. Appendicitis. An inflammation in the appendix called appendicitis. It's not commonly in elder people, found only in the children. And when you had the problem of appendicitis, a physician suggests to remove the appendix, that is surgical removal of appendix, that is appendectomy. This slide shows you the approximate percentage of lymphocytes in lymphoid organs. First column is lymphoid organs, and third is T and the B cells. Look at the first one, the thymus. We all know that from the thymus only T cells will be originated. So here it is 100% of T cells in thymus, and there is no B cells found in thymus. And look at the second one, the bone marrow. Bone marrow is the primary lymphoid organ where T and the B cells, both the cells are originated. So here the percentage of T cells is 10 and B cells is 90. As like that, the percentage of T and B lymphocytes present in spleen, lymph node and blood it is listed out. This slide shows you the components of lymphatic system. The first one is a lymph, and it is usually a clear colorless fluid. And the second one is the lymphatic vessels. Under the category, the capillaries, collecting vessels, the trunks, all coming under the lymphatic vessels. And lymphoid tissues, nothing but the malt and the galt. And the lymphoid organs, we are knowing the thymus, bone marrow, lymph nodes, tonsils. And now coming with the primary and secondary lymphoid organ differentiation. First of all, the primary is otherwise called as central, and secondary is otherwise called as periparal. And the second difference coming with the primary, and they are the sites where WBCs are generated, and secondary lymphoid organ are the sites where adaptive immune response it is initiated. And the third difference is about the proliferation differentiation. So the proliferation and differentiation of all lymphocytes, it is occur in the primary, and the lymphoid stem cells, it becomes functional in the secondary. In primary, it contains either B cells or the T cells, but in secondary, it contains both the T cells and the B cells. In primary, antigens cannot be entered, and here in secondary, antigens enters in, so that immune response elicited. And about the maturation, in primary lymphoid organs, the T cells and the B cells, it is maturated only in the primary, and in the secondary, the T cells and the B cells will be functional. That means the B cells interacting with the antigens, so that antibodies will be producing. And the primary lymphoid organs, it develops before secondary lymphoid organ, and secondary lymphoid organ, it develops after the primary lymphoid organ. And the next difference about the differentiation, the differentiation of lymphocytes is taken place only in primary, and here in secondary there is no differentiation. Instead of differentiation, interaction of immune cells with each other and antigen processing. And at last, about the anatomy of the organs, in the primary it decreases in size and strength with age, and here in secondary it increases in size with age. So that's all the difference between the primary and the secondary lymphoid organs. Thank you.